right, everyone, thank you for joining us. We'll give it a couple of more seconds for some other people to join us, and then we'll get started with today's webinar. Joining us, we'll give it. Okay. So hello everyone, thank you for joining us today. It's just right after 3 o'clock Eastern, so I'll go ahead and get us started. Welcome to the January 23 COVID Information Commons Research Webinar. This is our first session of the new year, so we're very excited. My name is Lauren Close. I'm the Operations and Communications Manager for the Northeast Big Data Innovation Hub here at Columbia University. I'm also a member of the COVID Information Commons project team. And I'd like to introduce you all to the COVID Info Commons, or the KIC as we call it. We are a COVID-19 research collaboration platform brought to you by the Big Data Innovation Hubs and funded by the National Science Foundation's Convergence Accelerator. The KIC brings together scholars from all over the country and around the world to share their research in the form of lightning talks. And today, each scholar that we have with us will engage our community directly, answering your questions about their work during a Q&A session at the end of the presentations. I should also note that we at the KIC are committed to providing a supportive and welcoming environment to everyone who works, studies, and interacts with our community at these public events. We encourage you to review our code of conduct for online programs. This includes information about reporting and accessibility features, so I will drop a link to those resources in the chat for you all shortly. But before I hand you over to today's fantastic group of speakers, I'd like to first introduce the Hub's Executive Director, Florence Hudson, who can give us some background information on the KIC.
Wonderful. Thank you, Lauren, and thank you for organizing these as always. I really appreciate it. So as Lauren mentioned, I'm Florence Hudson. I'm Executive Director for the Northeast Big Data Innovation Hub, which has been funded by NSF since 2015. And together with the other Big Data Innovation Hubs, where the Northeast, there's a Midwest Hub, South Hub, and West Hub in the US funded by NSF. We actually kicked off the COVID Information Commons in 2020 when NSF asked us to create a portal for people to easily be able to find NSF-funded COVID research. At the time, it was the RAPID Awards for Rapid Response Research. So when they contacted us initially, there were 32 NSF COVID Rapids. I thought this will be easy. And now we have over 9,000 awards in the KIC because we have all the NSF awards since then, and we've added the NIH awards as well. So we're delighted that we've had 2 awards from NSF, the KIC in 2020, the COVID Info Commons, and then this COVID Information Commons Extension Award, which we received in October 2021. The goal with that was to extend this to all the COVID-related research in NSF, to add the NIH-funded research, because after all, this is a disease, and we should be collaborating across the NSF and NIH researchers, PIs, and content. And also, the next steps in this award are to create an advanced metadata and data search and discovery mechanism around the world to bring together and to bring to light more of the research metadata and data that can be related to COVID, the ongoing challenges with COVID, the new pandemics we have to worry about, and the confluence of all of these diseases together as we move forward in this new world that we're in. So we're very thankful to NSF for their support. The COVID Info Commons, as I mentioned, when it started was just a small little thing. Now we have 9,045 NSF and NIH awards. We use the NSF and NIH APIs to monthly download the current research, the new research, and we use various mechanisms to be able to classify that. So you can see here that we have a faceted search that includes NSF Directorate or NIH Institute or Center, awardee organization, state or territory, PI name. You could put keywords like epidemiology, up in the top, or machine learning, and find research that might be pertinent to yours or pertinent to some of your ongoing thinking. You can also download a CSV file with up to 1,000 records at a time. And what you'll find in here is information that is available publicly through the NSF award site or the NIH award site, or if the PI has filled in a PI survey and provided additional information, links to the project website, collaboration opportunities, co-PI information, and things like that. And our goal is to help you all work together as we look to learn from what we've been through and try to plan for the future. We also have an annual COVID Information Commons Student Paper Challenge. We did the first one in 2021, which was just undergraduate. It went really well. So then we expanded it to undergraduate and graduate cohorts. And we actually just had those researchers, those student researchers, present on our inaugural Northeast Big Data Hub Student Research Symposium on January 27th. We had students from across the US and one in South Africa who won those student paper challenges. So we continue to help them use this information, to help them collaborate with each other. And then Lauren's going to be talking about some of our next steps beyond these lightning talk webinars we have for working groups that we're enabling around areas of interest to you as the researchers to help us continue to move forward together. So thank you all for being here. We really appreciate it. And we hope that you enjoy collaborating with each other today and in the future.
Yes, thank you, Florence. You're right. One thing I wanted to mention to the group today is a new initiative that we've begun with the COVID Information Commons in December. We now are supporting 3 new research working groups, which provide collaborative opportunities for COVID researchers. As you can see, some of the topics of these groups include education impacts, social impacts, and health outcomes. And we also have one on pandemic prevention and policy research. So anyone with an interest in these topics is welcome to join. Current members are from academia and also industry. We have students. I've included here a top-level overview of when each group will next meet. As you can see, the pandemic prevention group will be chatting next Friday. So we encourage you to join. You can find more info about these groups, including links to our past meeting notes on our website. I'll drop some of these details in the chat right now. So please feel free to share some information about these groups with your colleagues. And actually a handful of presentations today touch on some of these themes. So there's already a bit of overlap within the community, and we hope you'll join us at one of these meetings. I also want to mention, of course, that we have a dedicated student working group at the KIC. This project is spearheaded by 2 of our work study students at the Hub. This group meets at this point twice a month. We also have members from all over the world. Some people are based in Europe, others are in Europe, Africa, and Asia. It's really a wonderful international collaboration space for STEM students. And as with the researcher working groups, we encourage you to learn more about these future meetings via our website. So I'm going to be dropping even more links in the chat here. You can also, of course, join the conversation through our Slack channel, where we try to talk offline as well as in those calls. And our next meeting for students is this coming Friday, February 3rd at 12:30 Eastern. So we hope you or maybe some of your students may be interested in joining us. But without further ado, what I'd like to do now is introduce the speakers of today's COVID-19 webinar. We're going to be hearing from 4 fantastic speakers this afternoon. We have with us Mark Lurie, who's at Brown, Peter Pirolli at the Florida Institute for Human and Machine Cognition, Rebecca Powell at Icahn School of Medicine at Mount Sinai, and last but certainly not least, we have Jennifer Surtees, who's at the University at Buffalo. And so I'd like to kick off this afternoon's webinar by introducing us to our first speaker, Mark Lurie. He will be sharing his research on pandemic prevention strategies and analysis, a perfect match, as I was saying, for our pandemic prevention working group. But I will stop sharing my screen and let Mark go ahead and kick off those presentations. Thank you, Mark.
Lauren, can you give me a thumbs up if you're seeing my slides well? I see 2 thumbs up. Great. Good afternoon, everyone. Thank you for being here, and thanks to the KIC group for organizing this and other seminars. I'm honored to be here representing my team from Brown University. Many people from the School of Engineering, School of Public Health, departments of computer science, philosophy, and others, too many to list on the current slide, but if any of them are listening along today know that I appreciate all of their meaningful collaborations. I'm going to talk today about newish projects that's just started with NSF funding. Some people will be familiar with a relatively recent NSF mechanism listed here. And I'm going to speak about it a little bit, partly because our funding comes from this mechanism, but also two of the other speakers who are with us today will also be speaking about their project from the same mechanism. So the NSF asked us to think to the future, to think in a kind of 5 to 10 year span about knowledge, methodologies, data, that in that period of time could potentially be available that would put us on more solid footing to be able to prevent and predict future pandemics. So that was the call for proposals, was to have a grand vision of an area of intelligence that we could address that in 5 to 10 years would leave us on better footing to allow us to hopefully prevent the next pandemic before it occurs. Our study and our project is focused on all aspects of human mobility and social mixing. So why are we interested in mobility and social mixing? I'll give you 2 or 3 slides on that as some background. The first is the obvious statement that infectious diseases move from people to people. Therefore, how we move through space and time will be a major determinant on potential for a new pathogen to spread through populations. More importantly, knowing the details about how people interact will give us, in the future, more refined tools that'll allow us to implement more effective and more nuanced interventions. All of us remember back to the beginning of this pandemic when essentially the interventions that were implemented were those. Many of them were aimed at cutting our social networks, limiting the number of people that we interact with in order to slow the spread. And because I argue that we didn't at that time have really good quality information about how people interact, we were forced to apply a hammer to the situation, an unrefined tool that forced essentially all mixing to stop. If we are successful in the future, we'll better be able to map mixing that is conducive to spread and mixing that doesn't involve spread of new pathogens. And therefore, we can be more nuanced in the interventions that we propose in the future. As an epidemiologist, I think of this in another way as well. And that is, I'm sure many are very familiar with this epidemiological concept of the basic reproductive number, which really tells us the likelihood of pathogenic spread. And it tells us on average how many new infections will occur in a population when a primary case is introduced into that totally susceptible population. And R naught, the basic reproductive number, is dictated by 3 components. The first is how likely transmission is to happen between any contact. The second is how long is the period of infectiousness. And the third is about how frequently people who are uninfected come in contact with infected people. It's this last piece, the C shown here, that is really the focus of our work. In essence, the other 2 variables are largely biological variables that are going to differ depending on the new pathogen that emerges. But the human behavioral aspect is really contained in C here, and that's in essence what we focus our MAPS project on. I'm going to talk now briefly about the main components of our project, 4 main components, and then 1 overarching sort of proof of concept exercise. First, to articulate what our grand challenge was specifically, essentially we're asking how we can best use data on mobility and population mixing to inform real-time pandemic responses across a range of pathogens and under conditions of uncertainty while still balancing benefits, risks, and harms. To do that, we focus on 4 different areas shown here, and I'll speak briefly about what we do in each of these 4 different thrust areas. The first one relates to data. Clearly, there's some, though I would argue not nearly enough, data on how we interact in different places and in different contexts. So we're not the first people to collect or want access to this kind of data. What we're recognizing the existence of some data already, what we're trying to do here is create a catalog and make publicly available online a federated database that contains a multitude of different studies that focus on social mixing and mobility. We hope that this will be a resource for modelers, for pandemic researchers who are interested in incorporating social mixing and mobility into their study, to be able to go to one central clearinghouse and find available data, hopefully already in a format that can be used in their models. The second aspect that we focus on is around developing devices. So we work very closely with engineers here at Brown and biomedical engineers, trying to develop new technologies for measuring mobility, social interaction, and eventually biometrics. Right now, we're focused on a phone app, which I'm going to talk about in a bit. But there are potentially other devices and either other methodologies that can help us to understand people's movement. The third aspect that we focus on is around modeling and prediction. So the data that we collect through our apps and through our wearables, and the data that we store and process in the first thrust are meant to be to feed our predictive models. And what we're hoping to do here is to develop a library of models that are flexible enough to be able to respond to new pathogens that emerge. So we're looking at a variety of different models that incorporate social mixing and human mobility, and to make them, again, flexible enough so that they're not specifically COVID focused, but are able to be adapted to the epidemiological context for a new pathogen about which we don't yet know anything. Finally, a lot of our work is around ethics. And although it's listed as one of our thrusts, in a way, it really permeates all of the work that we do. To collect and catalog the kind of data that we're talking about means to possess people's private and confidential information. And so we need to be exceedingly careful about how we do that, what data we collect, how we use that data, and how we keep it in a safe and respectful way. We've just finished a week-long workshop where we brought together ethicists, public health folks, computer science people, and cryptographers to help us think about the technical challenges that we can address when using the kind of data that we're talking about, and doing so in a respectful and ethical way. So those are the 4 main thrusts or areas of work that our project is involved in. And I want to just speak now very briefly about an overarching almost proof of concept exercise that we're doing that brings together all of these different thrusts all at once. And the idea is that using the university where myself and my collaborators are based, that we would like to try to measure the entire social network at the university. So imagine if everybody at the university had downloaded our app, which is still in development. The app will use Bluetooth to measure who's in your vicinity. That is to say, it'll measure the interactions that you're having, the duration of those interactions, and the distance of those interactions. That allows us to build a dynamic model of the social network at the university. What we then want to do is simulate the introduction of a new pathogen into that network, and assign to the pathogen some specific epidemiological characteristics, whether that's the probability of transmission or specifically how transmission occurs, whether that's within 5 feet for 10 minutes or whatever those particularly characteristics are. And it allows us to understand not only how the pathogen spreads through the network under different epidemiological characteristics, but also to identify points of intervention. That is, things that we could change in the way that we mix and interact with others that could be effective in eliminating or containing the virtual pathogen. Our goal here is within the next 18 months to do a pilot within the School of Public Health. We've already been engaged in some deep community engagement, speaking with leadership, students, faculty, staff, potential users of this device about some of their requirements for the device and suggestions for how the device might look and feel, and also addressing concerns about data security and data privacy. We're hoping to get second phase of funding for this project. That would allow us to expand our work beyond the School of Public Health and to try to map the entire social network at the university. And beyond that, we're hoping eventually to add other measurements, including biometric measurements, potentially other measures that we haven't even yet thought about, that could be used for not only mapping and understanding social networks, but also to populate models that will allow us to predict and therefore prevent future pandemics. So that's all I'm going to say at this moment. I'm happy to answer questions and being involved in discussion about what it is that we're doing. I just do want to show this final slide here if anyone is interested in the workshop that I mentioned a couple of slides back, where we focused on privacy and ethics in pandemic data collection. And you can find some of the talks and slides there, as well as some of the background materials and daily summaries from that workshop. So I'm going to leave it at that, and I will stop sharing and turn over to my colleagues for the next presentation. Thank you.
Thank you, Mark. That's really very interesting. And to our audience, I'm sure you have lots of questions about this and the other presentations. So I just want to mention that we will, of course, be hosting a Q&A session at the end of today's webinar. In the meantime, if you'd like to submit your questions to Mark or any of our speakers in the chat, please feel free to do so or hang on to them for our live conversation. But I don't want to take any more time. I'm going to turn this over to Peter, who's going to be giving us our second presentation this afternoon.
Okay, are we good?
Looks
great. All right. Okay, great. Thank you. So the title of my talk is also the title of a proposal that we also put into the PIP program that Mark just mentioned. This is work that I'm doing with my co-PIs, Kathleen Carley, Christian Lebier, and Mark Orr. I'm at IHMC, and we've also involved researchers from Carnegie Mellon University and the University of Virginia. And our proposed grand challenge is really around the development of novel computational theories and models of information flow, human behavior, and the transmission and evolution of pathogens. And our work draws upon research from a variety of fields, mostly centering around the cognitive sciences. So it involves research on social and organizational systems, online social media analysis, machine learning, natural language processing, with a big focus on how to pull that all together into computational cognitive modeling of people. This is sort of an outline of the kind of work that we have been doing and the kind of center that we're trying to pull together. We collect data from a large variety of sources, including online social media and polling data that has been made publicly available. We're looking into the kind of research that Mark is talking about, where we want to employ mobile platforms to collect not only social distancing data, but also ecological momentary assessments of what people are doing and what they're thinking. And from all of these data, we're developing a variety of analytic methods to infer the perceptions, attitudes, beliefs, and intentions of people in different geographical areas. We use that to seed something that we call psychologically valid agents, which are based on a computational neurocognitive theory that I'll talk some more about. And the aim of that is to really try to understand the behavior response profiles of people in these different regions. That is, given the current state of the world, how are they social distancing? How, are they wearing masks? Are they making decisions to vaccinate or not, and so on? And those agents go into kind of synthetic population or agent-based model in which the main thing that we're trying to predict is the time dynamics of these behaviors. Mostly we've been focusing in so far on mobility, predicting mobility and predicting mask wearing. And of course, all of that is related to case rates and death rates, as Mark was indicating. What I'm going to focus in on is a very thin slice through this that focuses in on this idea of using computational cognitive models to make inferences about people's decision making. I'm going to focus on this because I think it's probably novel in this space. We are working with a computational neurocognitive theory called ACT-R. This is a theory that's been developed for some decades now. It is partly a theory of how the mind works, and it's also a theory of how it is implemented in the brain. So it covers a large variety, hundreds of experiments in cognitive psychology, but also makes predictions about the functioning of the brain. For example, fMRI data and EEG data that's collected as people do various kinds of tasks. It's been used in application in the development of cognitive tutoring systems. It's been used a lot in human-computer interaction. Underlying these models are a lot of very complicated dynamical equations that I'm not going to go into, but they cover things like how memory works, how forgetting works, how practice and habit formation work, and so on. And it's a unification of all these various aspects of cognition into a single theory in which we can do simulations. So for the purposes of this talk, I'm going to focus in on a subset of the mechanisms that are involved in decision making that come from this cognitive theory that are sometimes called instance-based learning or memory blending. And this is a particular kind of decision making that we think we frequently see in people doing real world decisions, where they are making decisions based on a generalization of their memories and experiences of previous situations. This primarily leverages the cognitive mechanisms that we know around memory, so how information decays in memory, how rehearsal increases the retention of those memories, how priming works, and it leverages a variety of cognitive mechanisms that are involved in what's called pattern matching. That is, how memories are retrieved in accordance to how they match the current situation. And also this mechanism called blending, which essentially generalizes over these experiences to produce kind of the best result for the current situation. And individual biases in decision-making result from the fact that people have different kinds of experiences and the way that these cognitive mechanisms actually work. And so to give you a thumbnail sketch of how the theory works in the simulation, the idea is every experience, every message goes into memory as what is technically called a chunk. And a chunk is just a unitary thing that captures the features of your particular experience or memory and stores that away. And of course, over time, you have lots of experiences that might be interrelated with respect to some situation or some decision. And at the time that a new decision is needed, you perform this kind of memory very quick memory retrieval that essentially generalizes and summarizes all that past experience based on the likelihood that it would be retrieved in this particular context, as well as the similarity and the intersimilarity of these experiences to the particular situation that you're making a decision about. So we use these mechanisms to model attitudes and behavior. And this is not very standard fare for how this this kind of modeling has been done in this community. And so this is a rather novel application to try to make predictions about people's attitudes and how it influences behavior. And attitude theory really comes from social psychology, and there's lots of attitude theories, but here's just a canonical theory to give you an idea of how it works. The idea is that your behavioral decisions, whether you're going to wear a mask or socially distance, are influenced by what are called intentions, and your intentions can vary in strength. And those intentions are related to your attitudes towards the likelihood that an outcome is going to happen and the value of that outcome. It's also influenced by subjective norms. What are other people doing? What do other people think about what you're doing? And it's also related to what's called perceived behavioral control, or what's often called self-efficacy, which is your belief or confidence in your ability to do something. So those things combine to make predictions about attitudes and in the implementation of behaviors. We implement this using this set of mechanisms called instance-based learning, where the basic idea is you have a bunch of experiences that are positive around some behaviors, sometimes negative. You're getting social information, you're getting messaging information. And at the time that you have to make a decision to to execute a behavior or not, that all those pieces of information and memory and experiences are being combined into that particular decision to, for example, wear a mask or not. And here's just kind of a toy simulation just to kind of give you a sense of the impact of messages and experiences on intentions and decision making. So this is just a toy simulation in which we assume that there's no real value that you place on mask wearing, but then you get messages at discrete points in time that say it's highly valued. Those messages get stored as chunks, and they influence your inferences about the subjective value of wearing a mask. And if the cumulative effects of those messages and the timing of those effects of messages impact your overall intention and your expectations, and then that has an impact on your decision making about whether to execute that behavior or not. Just to go through another thing that influences behavior, this notion of self-efficacy. The idea is that you build up confidence every time you positively execute a behavior. When you think you may not have enough confidence or you may have some difficulty, you might put additional intentional effort in. And so over time, as you do something like wear a mask or socially distance, your self-efficacy builds up, the amount of additional intentional effort that you have to put in goes down, and overall the probability of that behavior goes up. So how does this all combine in some phenomena that we see in things like mask wearing in reaction to COVID? One of the things that we have been modeling specifically is this idea that your awareness of what's going on in the pandemic around you and the messages that you're getting through mass media and social media have an impact on your awareness of the state of the pandemic. And then that modulates your behavior, which then in turn modulates the transmission rates, this effective transmission number. However, there's delays in how this all propagates. So there's a delay between the point in time that people get infected, that the symptoms become apparent, to fatalities, and then to our awareness of all of that. And that results in a kind of oscillatory dynamics that you can see in the data themselves. So this is the effective transmission number in 10 carefully selected states here. And what you see is that this is over the first 3 waves of the COVID pandemic, that there's this huge spike in transmission that is then brought down and kind of oscillates around 1. And our simulations can reproduce that kind of oscillatory behavior. In the lower right corner there is kind of a phase space diagram where the infection rate at time t is then related to the probability of people wearing a mask, the idea that people wear a mask in reaction to their awareness that things have gotten worse, and then they may back off of that when they think things are getting better. And one way to think of this is it's kind of like a pendulum is swinging back and forth, and that as the pendulum swings in one direction, there's a behavior that kind of pushes it back in the other direction, and then when it swings in the other direction, there's a behavior that pushes it back towards the center, which causes it to kind of oscillate around this effective transmission number of 1. Now, when you look at the data, it's actually a little more complicated than that. So this is the same kind of phase space diagram where we're looking at the RT values at one point in time related to the RT values about 2 or 3 weeks in the future. And you do see this kind of oscillation around 1, but there's also this kind of spiraling where it kind of is also moving upward a little bit. And if we look at the actual relationship between transmission rates and people wearing masks slightly ahead in the future, we see an even more complicated picture where there is this kind of oscillation that as RT values go up, people seem to wear masks, which brings it back down, and it oscillates back and forth. But it's also overall increasing, again, over the 3 waves of COVID, such that people's reactions by the 3rd wave are that when RT values go up, they almost immediately start to wear masks and more people wear masks. And we believe this is kind of a reflection of the fact that there is this kind of a learning effect that people are exhibiting, which fundamentally kind of goes back to this notion that people are building up self-efficacy, and they're also building up some habits around how to do that, which is perfectly captured within the model. So that's just one of the phenomena that we're looking at. We're looking at many other aspects of how people behaviorally react to information in their environment. This again is just showing these data in a slightly different format. The top 2 graphs there show the RT values across 3,000 counties over the first 3 waves of COVID. So the top left there is the first and second wave, and you can kind of see that they all center around 1 and are basically the same across those 2 waves. And you see the same pattern as you go from wave 2 to wave 3 in that top right chart. But then if you look at the charts below, this is the percentage of people wearing masks over these 3 waves. And you can see as you go from wave 1 to wave 2, mask wearing goes up dramatically, and then a little bit more when you go from wave 2 to wave 3. Okay, so we're doing additional work, as I said, predicting other kinds of behaviors. We're looking at mask wearing, social distancing, and now we're turning towards an analysis of vaccine attitudes and decisions, as well as people's attitudes towards alternative treatments besides vaccines. So we're looking at analytics around vaccination discussions in mass media and social media, segmented geographically across the United States, using a variety of machine learning and natural language processing to identify these beliefs, attitudes, and also very interested in credibility judgments about how people perceive the sources of this information, and then pulling that all into these psychologically valid agents that are built in ACTAR. And that's it. Thanks.
Thank you so much, Peter. That was really helpful. So fantastic. Right now, I want to turn the webinar over to our next speaker, Rebecca Powell. She will be discussing her work on COVID antibodies and breastfeeding.
Everyone. Okay, so yeah, you can see thumbs up. You can see my screen. I assume so, yes. Okay. Right. So I'm a human milk immunologist at Mount Sinai in New York City. Today I'm going to talk to you about our work on the human milk immune response to SARS-CoV-2 infection. So just because not everyone's coming from the same background, I'm going to give a little bit of background on antibodies and other parts of the immune system in general. So antibodies are a protective protein produced by B cells in response to the presence of a foreign substance, and they recognize and latch onto the foreign substance so that they can be removed from the body. And when this foreign substance enters the body, the immune system recognizes it because its proteins differ from the self, and any antibodies that might react with self are, at least in theory, not present and eliminated as part of the development of the immune system. So those proteins from a foreign element like a virus are very different from our own proteins. And so an antibody has two sides. It has the variable side where you see the V's here. That's what contacts the pathogen, like SARS-CoV-2, COVID-19. And those are different for every antibody, is a little bit different in its variable region, and that is how they recognize a foreign protein. And then this other side is the constant region, or the FC region, and that's what will mediate some other antiviral activities and bind to receptors on some of the cells of our immune system. So there are different flavors of antibodies. I'm mainly going to be talking about IgA today because IgA is about 90% of antibody in milk. But in serum, so in our blood, you mostly hear about IgG. So that's what's dominant in serum. And then there are other flavors I won't get into today, but just so you know, there's many different types of antibodies, and it's not so simple just to say antibodies and be done with it. So there's many types. So in human milk, antibody, as I said, is about 90% IgA, mostly as a dimer, meaning two IgAs bound together. And most of that is going to be in secretory form. So secretory antibodies are polymeric. So that means they have different types of proteins combined. And they're, in this case, a dimer, which is mostly how you find IgA in milk, is going to be bound together by another protein called J chain that's made by the B cells, and then wrapped up in secretory component. So this is a little cartoon of that. There are two kinds of IgA. I'm not going to get into that today, but they're structurally a little bit different, and IgA1 is dominant. So here you see a dimer, which is complexed by J chain, which is made by the B cells, and that's mostly what you find in milk in terms of the total composition. And then it gets wrapped up as part of its secretion into milk in this secretory component. So secretory component is critical for the protection against degradation in harsh mucosal environments like milk, like the baby's oral nasal cavity, and especially the GI tract. So we evolved to have this highly protected form of antibody in our mucosal fluids because they are relatively harsh compared to serum. So what's interesting is that one thing we're exploring in my lab is that secretory IgA derived from milk, because you can get milk in large quantities, as a therapeutic class of antibody against COVID or other pathogens, may be highly efficient when you need those antibodies to be durable in those harsh mucosal environments like the respiratory tract, the GI tract. So secretory antibodies, you don't have to get too wrapped up in this diagram, but as I said, and for those that are interested, the way that they get their secretory component is that actually when the B cells transit to the mammary gland and they secrete that IgA dimer, as it's being pumped into the milk, a piece of the receptor that pumps the IgA into the milk comes off, and that's secretory chain. And then that is what wraps up the IgA. So we also have IgG and other forms in milk. IgG is about 2% of the total, and that's going to generally come from the serum, coming in a relatively passive manner. The B cells that ultimately produce milk secretory IgA actually originate mostly from the gut, from the gut-associated lymphatic tissue. And so this is known as the enteromammary link, and you can imagine this is a very important evolved mechanism to help babies survive in an environment as we were evolving, or in low-income settings today where modern medicine is not available, and whatever the lactating person breathes in or ingests in the form of pathogen, then an immune response is raised in the gut predominantly, and then B cells from the gut transit through the lymphatics to the mammary gland and thus make antibodies that are highly durable in the milk that actually protect the baby from those very same pathogens and stop the baby from dying from those pathogens. What is passive immunization? So when a baby or child gets human milk, it's not like they're getting vaccinated. It's not like they're getting infected themselves, but it's what we call passive immunization. So this passive protection works by coating the mucous membranes of the baby's mouth, upper respiratory area, and digestive tract, and it provides a layer of protection that may stop cells in those areas from being infected if the baby's exposed to virus, or it may mitigate that infection, so slow down viral replication. So this effect is temporary, and the antibodies would be expected to be degraded or washed away within a few hours. So this has to be replenished with every feed. Now, babies get their milk very frequently, so this should not be a problem. But the effect is going to be dose dependent. So your exclusively breastfed baby who's not getting any other food or fluids is going to have the longest lasting coat of milk antibodies. And then a toddler who's also eating solids, it's not going to be that significant of an effect, relatively speaking. Now, these milk antibodies do not pass through the baby's digestive system into the bloodstream. You would not detect them in the blood. They are a coating of the mucous membranes. So our COVID-19 studies, we have two of them. I was NIH funded with an R01 for our infection study in 2020 as part of the emergency funding. And we also have a vaccine study that I'm just going to mention at the end of my presentation. So for our infection study, the participants had a PCR-confirmed SARS-CoV-2 infection, or for our vaccine study, they were vaccinated, but that's a separate study. We asked for about 30 mils or 1 ounce of milk per sample, using whatever pumps these participants had at home and were comfortable with. The milk was pumped at home according to a schedule that we requested, labeled and frozen at home, and then picked up originally by me during the shutdown, and then later by courier or shipment by Milk Stork, a milk carrier company. And then this was designed as a longitudinal study. So milk was obtained originally 3 to 6 weeks after infection. And then we asked participants to continue to pump monthly samples for as long as they were able. So initially, to show you the results from our COVID infection study, in a subset of samples, we have many samples. We have hundreds and hundreds of participants. In this part of the study, we screened undiluted processed milk. So we just centrifuge the milk to remove the fat and the cells, and we keep the skim milk fraction, which is what we test. So we looked at 75 COVID-19 recovered participants, and also we used 20 pre-pandemic milk samples from other studies to form a positive cutoff in terms of antibody against the COVID-19 spike. We use an ELISA, which is a very tried and tested method to look at the response of antibodies in a solution of any fluid against any protein of interest. So we get recombinant spike protein, we code it down to a plastic assay plate, and we essentially just look at the response of the milk against that spike protein, and we can quantitate how much antibody is there. So looking at 75 people, and this dotted line being our positive cutoff, which is based on our pre-pandemic control background levels in this ELISA, we found that just about 90% of milk samples obtained from these COVID-19 recovered donors had significant levels of SARS-CoV-2 specific IgA. This is an absolute positive cutoff level. So if you have samples from before an infection, which we did not at the time for this subset, and you could compare the relative increase for particular individuals, it's actually much more like 100% of people have a response after infection. But our absolute cutoff is pretty stringent. So in that sense, it's about 90% past the absolute cutoff for positivity. This initial screening, 40 of the spike positive samples were further titrated, so serially diluted to find the binding endpoint titer. So this is an assessment of antibody affinity and or quantity. So the way you do that is you basically look at what dilution of milk gives an optical density value of 1, so the amount of light in this ELISA assay. And then you can see each of these is the dilution curve. You can see every milk sample, although positive, looks very different because the quality of the response differs. And so what we found is that of these 40 samples that were positive on the screen, 95% of those had a positive endpoint titer, so they were of high quality, and about half of those were what we consider high titer, so a high quality or quantity of antibody in the sample, which is about 5 times the endpoint titer cutoff. And about 95% also had spike-specific secretory antibody, which of course we're very interested in because that is the most durable class of antibody in milk. So when we do our secretory antibody ELISA, we look for antibodies, we look for secretory components. So that could be on IgA, it could be on IgM, which also has a secretory form. It could be some other strange artifact like just free-floating secretory component, but that's a lot less likely. But just because we measure IgA doesn't mean we know it's secretory IgA until we also measure secretory antibody and compare those data. So what we also found was that about 75% of these samples did contain spike-specific IgG, which is the dominant form in serum, but can also be detected in milk. But it was very low, and as you can see, only about really 1 passed our high titer cutoff, and it was generally a low titer IgG response. But it's hard to detect IgG in milk because overall very little is in the milk. What was very important was that we found that IgA and secretory antibody titers were highly correlated, which strongly suggested that most or nearly all of the IgA was in secretory form, which is very important for potential protection of the baby. And we also performed neutralization assays, which looked at the ability of these antibodies to actually block SARS-CoV-2 infection. This was using a pseudovirus, which is a harmless virus that's been made to have the SARS-CoV-2 spike attached to it. And so we can measure how well the antibodies block infection. And what we found was that there was potent COVID neutralizing activity, which strongly correlated with the amount of secretory IgA in the sample. And then finally, what we found more recently, which is very important, is that the milk secretory IgA infection response is very durable over time. So when we have time points up to 12 months post-infection, what you can see is that there is hardly a significant change at all in these 20 or so people that we have looked at so far. And you can see that when we do the statistical analysis, there is no change between these groups at any time point in terms of the endpoint titer for the secretory IgA. So this is a very, very durable response over time after infection. So to summarize, the post-COVID-19 infection response is secretory IgA dominant, which really is a classic mucosal infection response. The secretory IgA is neutralizing, and it's very, very long lasting over time. Now, I didn't show you, but I'll just tell you that the post-COVID-19 vaccine response is actually IgG dominant. It's a classic intramuscular injection response. It does not follow this mucosal pathway at all. And the adeno-based vaccines like J&J and AstraZeneca actually had very poor responses in milk, very low titers, and very, very low secretory antibody was actually found for any type of vaccine. And it was very limited durability over time. So a strong contrast to the mucosal infection response. The intramuscular injection response was really different and not very long lasting. So what this, really tells us is that vaccines that elicit a potent secretory IgA response, like your classic mucosal infection response, those vaccines are really needed. And that's one of the overarching goals of my lab, is to design those vaccines with the lactating population in mind. And we have some preliminary animal studies towards that goal right now. I'd like to thank everyone in my lab, the Kramer lab for the spike protein, Medela for initially supporting milk shipping costs by Milk Stork. I'm supported by the NIH and, of course, our milk donors. Thank you.
That's great. Thank you so much, Rebecca. It's fascinating. But I'd like to finally turn us over today to our last speaker, Jennifer, who's going to talk to us a little bit about her pandemic detection research.
First, I have to unmute myself. Okay. Good afternoon, afternoon, everybody. Thanks for being here, and I am excited to talk a little bit about what we are doing. This is our PIP, or Pandemic Prevention Project, funded by the NSF, that Mark talked about earlier, and so this is another one in this mechanism. My name is Jennifer Surtees, and I am at the University of Buffalo, and I just wanted to start by sharing with you what our grand challenge is for pandemic prevention. So basically, we are the Center for Ecosystems Data Integration for pandemic early warning systems. So our grand challenge is to basically develop a number of streams of data at different levels, to develop this that we can integrate to develop an early warning system for outbreaks and pandemics. So the idea is to integrate multiple diverse databases that encompass the viral ecosystem, which is derived from wastewater samples and patient samples. The human ecosystem based on big data, GPS data, cell phone data, as well as community level ecosystem information based on talking to the community and different members of our community. And so we see this development of data and integration into an early warning system combined with communication of our findings, solicitation of feedback from our community members, and generating information from our community members to integrate into our early warning system as this feedback loop mechanism for generating this more robust ecosystem data or early warning system. And so we have as a goal to develop this early warning system, but also as an equally important goal is to engage with our communities and build real partnerships, not only for the generation of data for our early warning system, but also as a mechanism to have real partnerships and a trust relationship with our various community members. Because it doesn't matter how good our early warning system is if when we do issue warnings, the community doesn't respond. And so building that trust back is really critical. I'm not going to go through this entire slide. I just want to share with you the fact that this is a highly interdisciplinary group and a growing group from across the university, across the disciplines, ranging from genomics to mathematical modeling, machine learning, engineering, virology, history, disability studies, chemistry, etc. And so this is a terrific team. And outside the university, we have partnerships with different healthcare systems, New York State. We're part of a New York State whole genome sequencing consortium at the moment, and the Buffalo and Erie County water supply. So we have this fantastic network to help us generate and integrate and implement the data that we're talking about. And so really we're trying to use our region in Western New York. So again, I'm in Buffalo and we're in Erie County. So we're trying to use our region as a community lab. And so to generate database about our microbial ecosystem, what's happening in terms of the viruses and microbes that are present through both wastewater, which I'll talk, and individual patient samples, and I'll talk more about that in a few minutes. And also understanding our human ecosystem, our community relationship and capacity building. We, through a community advisory board, the development of surveys and focus groups, which I will talk about in a little bit, but also data mining and GPS monitoring and GPS building and network building for the purposes of modeling. So developing these different types of data sets at the microbial system level, the human level, and then putting them into this predictive modeling hub based on mathematical modeling and machine learning. And then having that output go out to our community members, our stakeholders, as well as to feeding back into our analysis of our ecosystems. And really critical to all of this is the communication to our stakeholders, to our community members, communication of the science, communication of risk. What level of risk are we at? So what underlies a lot of what we do is sequencing. And so I just very quickly want to orient you to SARS-CoV-2 sequencing and then beyond to other microbial sequencing approaches. And so I just want to orient you to what we're looking at when we look at these phylogenetic trees. And so basically we start off with an original SARS-CoV-2, the Wuhan virus, if you will. And then over time, as the virus spreads, it accumulates mutations. And so we get these different lineages and variants of concern ultimately. And so as we get changes, we get the branching off of this phylogenetic tree. And so in this particular case, we start with this blue sequence, but then on this clade, it's accumulated another new mutation, this orange circle, and then another mutation, this green circle. And so these two individuals, A and B, have the same lineage, and it's this combination of mutations. Lineage C is similar, but instead of having this green circle, it's got this red circle. And so while it's closely related, it is distinct from lineages A and B. And similarly, we have this other branch point where we have, again, related lineages, but with some differences. And so what we've done here in Western New York is, and people have done this all over the world, but this is looking at Western New York. This is our phylogenetic analysis of what we've seen throughout the pandemic. And so we've been able to build these phylogenetic trees to really assess what's present in our communities. But what we've also done, and this ties into our modeling, what we've also been able to do is look more closely at the different lineages to do a cluster analysis. And so by taking, in this case, all of the alpha variants that we have identified in our region and clustering them based on the similarity of the sequences, using those phylogenetic trees, we see distinct clusters. And so the more yellow it is, the more similar the alpha lineages actually are. So we see these distinct clusters labeled 1, 2, 3, and 4, which are all alpha variants, but then have background mutations which make them distinct. And by looking at these differences, it suggests that there are multiple introductions into our community, well as community spread. So different versions have been introduced, and once they're introduced, they spread to other people. And this is just shown, you can see this in these phylogenetic trees. What these arrows are pointing at are actually unique mutations that are actually only ever seen in Western New York, which is kind of cool. The other thing, we can actually use the tracking the spread of SARS-CoV-2 to actually do agent-based modeling, which I'll talk about in a second, to actually understand how these different versions of SARS-CoV-2 move around and in our community and model that movement based on some of the human data that we will also integrate. One of the things that we did notice, so this is actually looking at the different lineages of SARS-CoV-2 across New York State. So these are the different regions within New York State, and the different panels represent the lineages over time, in this case in 2020, in these different regions. And if you just look at the color patterns, you can see that these, and actually we also included Ontario because it's nearby to Buffalo. But if you just look at all of the different patterns, the color patterns in these different regions, you can see there are really significant differences in the patterns of which version of SARS-CoV-2 is present, even in regions that are very close together. And this was even more evident when we look in our 2021, and I'm not going to show, but this is also true in 2022. So we think we can use this distribution and variation in distribution over time as a way of modeling the spread of SARS-CoV-2 throughout the state, as well as within Erie County over time. And so this is just again showing a time-lapse snapshot of the Alpha variant in New York State, where it was localized and when, with the more yellow being the more prevalent. And you can see it starting out in downstate and Eastern New York, and then gradually spreading. But the timing of this variant is distinct depending on what part of New York you're in. And this is just actually showing, this is just a GIF of showing the Alpha variant moving and around New York State over time. And this is just the New York variant to compare. And you can see differential migration and differential movement of these two lineages over time. And so by incorporating things like traffic patterns and developing agent-based modeling, we're doing network analysis across New York State and within Erie County to understand the movement of the virus through time. The other thing that we've been doing, and this is, with work with our engineering collaborators Yin Yi and Ian Bradley, who over the course of the pandemic were tracking levels of RNA in our wastewater. So this is just a cartoon of wastewater, collecting wastewater data. So this is our wastewater, and then these are the SARS-CoV-2 viruses that are present in our wastewater. So over the course of the pandemic, they looked at levels of RNA as a function of time. And then about a year and a half ago, we actually developed techniques for doing sequencing of SARS-CoV-2 in the wastewater as well. And so this is just showing patient samples, individual samples. And again, the colors are different lineages of SARS-CoV-2. And this is as a function of time on the x-axis. And when we look at 4 different sewer sheds, we can see that the virus type itself is similar among the sewer sheds, although there are some differences. But the patterns are actually different than what we see in the patient samples. And so the wastewater samples is actually a pooled sample of everything in our community. And so it's really interesting to look at the differences between the patient and the wastewater samples. Patient samples being a little bit more biased in terms of just who's actually getting sequenced. And so, of course, the wastewater can be used to monitor for different known and novel pathogens. And so this is what we're actually doing now. And of course, in order to really do this in a meaningful way, we need community cooperation and trust. And so now I'm just showing you the same diagram, but here I've added in a whole bunch more squiggles and triangles to indicate that we're not just sequencing COVID anymore, we're sequencing all of the RNA that's present in the wastewater after depleting for mRNA and rRNA so that we can enrich for some of the microbial and particularly viral RNAs. And the idea here is to develop a longitudinal picture of the viral and microbial ecosystem over time so that we can get a sense of what's normally there so that if it's disrupted, we can notice, and then we will notice and start looking for it in more detail at what might be present. So this is a way of detecting not only known pathogens, but potentially unknown pathogens as well. And one of the other things that we're really excited to do is to incorporate mass spectrometry to detect pharmaceuticals in the wastewater as well, because it looks like the uptick in ibuprofen and that sort of thing actually precedes an uptick or a spike in cases. We're also looking at the community level to really try to understand what biomarkers, what key questions do we need to ask our community partners in order to feed in modeling and anticipate an outbreak. So we're collecting oral histories, we're doing focus groups and interviews with different communities across Western New York. This involves a discussion of community concerns and interests, including the ethical issues that were mentioned earlier. And we have a strong community advisory board as well to help advise and to help us with our communication. And again, this data gets fed back into our models. So I also mentioned the importance of community engagement, and we have actually a really robust track record and set of partners to do community engagement, both in our K-12 schools and within our community, more broadly speaking. And these are just some images of some of the community engagement events and activities that we've done over the past 7 or 8 years. So we're leveraging those partners now, as well as a partnership with the Buffalo Museum of Science, to do a lot of community partnership building, hands-on type research. And this is one of our first sort of demonstrations for community engagement at the Museum of Science, where we have this demo of what wastewater actually is and how we collect all of those samples from within a neighborhood in order to get a big picture, a broad picture of what's going on at the viral level and the microbial level in our communities, and how we use that to predict and hopefully prevent pandemics. So I will stop there.
Wow, thank you so much, Jennifer. That's fascinating. And it reminds me a lot, I will say, of some work that's been discussed on wastewater in one of the working groups that I noted earlier. So I'll touch base with you on that at a different time. But I want to conclude everyone's presentations this afternoon by thanking all of you for sharing your research with us today. This is a really fantastic setting. We're so thrilled to be able to feature your work. And for the audience, let me say that each of our presentations will also be made available on our website, through our YouTube channel later this week as part of our Accessibility and Spanish Language Resources Initiative. This is a project that we're really proud of at the Northeast Hub. With the help of our students, we actually transcribe each of these monthly webinars, so individuals with various accessibility needs can either read about the research in English rather than listen to it. We've also translated those texts into Spanish, and the aim there is to make this rich content available to as many people as possible in different formats. So we really want to increase the diversity of our KIC community, and we're really grateful for all of your support on that front. So you can learn more about this and other initiatives on our website. Also, if you're interested in helping our team build out those features, please do reach out to us. We're always looking for more members on that team. And before we segue into the Q&A session of today's webinar, I do also want to briefly plug all the different ways that you can stay in touch with the COVID Info Commons. As you can see, you can connect with us through our website. We have a newsletter that I edit, so please read it. We also have Twitter, Instagram, Slack, LinkedIn. There's a bunch of different ways to stay in touch with this community. You can also reach out to us directly through our email address at the bottom of the screen. I will be sharing all of these links in the chat, so please peruse our channels at your own pace. Keep an eye out for our communications going forward about our next KIC webinar, which should be happening in April of this year. So now that I've done a little bit of admin, I will step back and really put the speakers back on the podium today. We have some great questions coming in from the chat. It seems like there's a couple things already happening. Miranda had a few questions for Rebecca. Let's see, Miranda, do you want to ask your questions directly, or did we touch on your points already? Oh, there she is.
I'm fine asking. Thank you, Rebecca. That's some fantastic work. One of the things I was really interested in, I mean, the logistics of that type of study and all the samples, and as you said, you had hundreds of participants. So I don't know what that means in terms of total number of samples that you had to process, and the fact that they were essentially all hand collected, is amazing. So I was wondering if you could just comment on some of the actual logistic parts of kind of, milk would seem to me to be a relatively fragile sort of medium that you had to kind of deal with. So
yeah, I mean, I think in some ways it can be, but the way we did it was, and the way that we still do it, so people, they pump it home, they freeze in their home freezers, and such participants are somewhat accustomed to properly freezing their milk, like they don't want it to thaw or waste, so they don't keep it in the door of their freezer, they make sure it stays frozen. If something happened at home with their freezers, they were supposed to, they would tell us, so there, was occasional issues like that where we could not use the samples, like if their freezers broke or something like that. As time went on, we would collect samples more in batches from people when they had, an adequate number. We still use Milk Stork, which their shipment mechanism it's like orchestrated through FedEx, but they have this special cold box that the donor activates at home, and it maintains the milk frozen. So essentially it comes from the freezer. We suggest that these 1 ounce samples are frozen flat. A lot of people have like ways of flattening these bags in their home freezers, because they do that anyway. And then we could fit maybe like 6 or 7 of them in these freezer boxes, and then they're picked up by FedEx. So generally, even in the worst summer situations, we would receive the milk still very, very cold. Most of the time it was still frozen. If it was definitely still frozen, and we would sometimes thaw and process it then anyways, but often we would just store it. So we would maintain that temperature. If it had already thawed but was cold, then we would just process it then. And on the rare occasion where obviously something had gone wrong, we couldn't use the sample. So if it was really like room temperature or worse or exploded, I mean, occasionally bad things happen, then luckily those were rare. So yeah, I mean, that's pretty much how it was done. So it can definitely be done.
Wow, it's quite an undertaking. Thank you.
Yeah, thank you so much. That's a great question. I have another question that came into me from the chat, and this one is for Mark. This is a long question, so bear with me as I read. How are you thinking of including potential social determinants of health implications and understanding or addressing the challenges of data capture and use with a broader population, including maybe, for example, underserved constituents with this type of approach? And can that be done through an institution like Brown with your pilot, or through a broader community engagement? Oh, Mark, I think you're on mute still.
Thanks. Sorry. Appreciate the question. Yeah, I think often when we think about sort of digital approaches to health, we are reminded that people don't have equal access to a lot of the technologies that we're talking about. So I think that's really important. And so in that way, what we're doing at Brown, or what anybody does when they're trying to measure a particular set of social interactions in a setting, is that they're shedding light on that particular setting. And so I think in a way that's why our Brown exercise is a kind of proof of concept to really test some of the ethical, not only ethical, but also, technical issues related to app development, user interface, things like that, to make it more usable. But probably a bit of an incoherent answer to a long question, but I'm happy to keep communicating with you or anybody else. Thanks.
No, thank you. That was a very cohesive answer to a complex question. Thank you for that. Peter, I have a question for you as well that came in. This one is also broad, I would say. Now that it is expected that the Biden administration will effectively end COVID health emergency declarations beginning this May, do you see that impacting the behaviors that you were talking about in the US?
I'm not exactly sure how it would. I mean, as far as I understand, the various emergency powers at the local and state and federal levels, were mainly utilized up through 2021, and now a lot of the the emergency powers are really being directed to kind of mopping up on more economic issues. Having said that, one of the things that's kind of very interesting in the data themselves, that I think really deserves a lot more study. And it's not just in the United States, but it's internationally, is that people's behavior in response to things like mandates and lockdowns is actually much less than you think, in the sense that things like social mobility and mask wearing, et cetera, seem to be responsive before government interventions. And this is not just characteristic of this pandemic. It's been remarked on that, if you go back through time, people very often sort of ran to the hills long before the virus ever showed up. So generally, at least as far as kind of, non-pharmaceutical related behaviors, people seem to change their behavior long before government mandates, et cetera, come into being. And so trying to understand a little bit more about why people are making those decisions and what information is having an impact, what social factors are determining that, seems to me to be kind of like a big thing that's unexplored right now. And I think kind of points back to the issue of how important it is that that the information flows and the credibility of information sources and the factual material that's being delivered to people really is, as crystalline as possible. So I guess that's my response to that.
Very interesting. And Miranda, I saw your hand up. Do you want to ask a follow-up question?
Actually, I had a question for Jennifer. And great to see the wonderful work being done here in Buffalo. But one of the things I was curious about with the I think the whole wastewater monitoring thing is fantastic, but do you have sufficient time, sort of fine resolution there, to be able to determine approximately how many sort of founding events, like, when a particular variant might appear in the community and whether it was brought in multiply or one person spreading or those types of, does the data sort of provide that type of information?
It depends. Yeah, maybe. So for the patient samples, I think it's easier because well, there are limitations there too, but for the patient data, it might be a little bit easier because you have some dates associated with them, and we have zip codes associated with them. For the waste, we actually sample twice a week. We get samples twice a week, and it's like a 24-hour sample that is collected from each sewer shed twice a week. So it's not terrible resolution, actually. And we're generating a lot of data. And I mentioned this New York State consortium where we initially started with patient samples, but it's expanded to wastewater now. So New York State is really invested in wastewater sequencing, not just of COVID, but of other pathogens as well. Which is really exciting to see. And so, and it's really fun to be part of that and part of the development of that. So I guess the answer is I don't know. And that's one of the things that we're trying to figure out is that, what is the resolution? How much can we actually learn? And then can some of the modeling that we build based on the patient samples and that sort of thing be sort of integrated into the wastewater as well? But yeah.
Great, thanks.
Sure.
I want to make sure that I'm opening up the floor to anyone else in the audience today, or any other questions from the group. I have others, but I don't want to step on anyone's toes. Okay. Well, one question I wanted to pose to the group of you, the presenters today, and this is actually in the spirit of the answer that you just gave, Jennifer, about there's still being work to be done, or there's still spaces to be explored. Is there anything about the research that you all have done that surprised you maybe, or that you weren't expecting? Very open question.
I can start. I described this exercise that we're trying to do at Brown to get people to adopt an app that would measure social interactions. And although we haven't piloted it yet, our early work really indicates something that surprised me is the lack of reluctance of people to accept and engage with an app like that. Young, students in particular felt like they're being surveilled in so many ways that this is actually a useful use of this data, and so they were interested and wanting to contribute. So that was a surprise. I'll pass it on to someone else.
Fascinating. Yes, Peter, please.
I think one of the surprising and probably most ephemeral things is this notion of trust is one of the key factors that seems to determine how things like pandemics proceed. So there's some papers that kind of do a post hoc analysis of how prepared we were for the pandemic. And after going through so many different variables, trying to understand why some countries did better than another, it turns out that that the trust in government institutions or traditionally social institutions is one of the key determinants of how different states, different countries responded to the pandemic. Similarly, the related notion of credibility, all sorts of information flowed about the pandemic, and people based their decisions on that. And one of the key factors, again, is what was the perceived credibility or trust of the sources of those information that determined whether people chose to do one thing versus another. And I know it's a very ephemeral idea, but it's kind of just remarkable to me how the entire edifice of public response seems to boil down to how much trust individuals have in sources of information, public institutions, et cetera. So having said that, policymakers or decision makers have this continuous thing that they need to be aware of. It's like, wouldn't it be great if you could sort of have a little trust meter that says, if you say this or say that or do this or do that, that it's going to change the credibility in the next thing that you say. And it's, just to expand on that, I mean, it's not just pandemic response, but like the entire financial institution relies on various forms of things that are whether you trust that the money that you've got in the bank is going to be there tomorrow. And so this, whatever it is that determines those perceptions, seems to have like an enormous impact on how people respond to the messaging and the decision making that is done by public officials. So that was, to me, kind of like, I guess I sort of knew it, but it's just been amplified by looking at the data.
No, that's fascinating. And Jennifer, I'm sure you also encountered issues of trust and credibility in your research as well. Yeah.
Yeah, trust is something that we've been working on for a long time. I mean, our initial stuff with community engagement is on genomic literacy and trying to having people understand genomics and how, what it means and why it's good, why it's bad. But I wholeheartedly agree with the trust, and that's one of the things we're trying to do is build trust. But that ties in because our approach is really a fairly local approach and a regional approach. And I think what maybe surprised me is the importance, or has struck me, is the importance of the regional in this pandemic response, the importance of knowing what's going on in our region with our communities in order to really effectively respond. It doesn't matter what's going on in New York City from the perspective of Western New York, because it's different and we're different. And so I think that regionality and the importance of that is something that has struck me.
Well, that's fascinating. All these new themes. Rebecca, is there anything that stuck out to you that was surprising or maybe unexpected in your research?
I was pretty surprised by how durable the antibody response in milk was over time, because, nobody's really been able to study this, in the context of any infection. or vaccine. There are, I'm not the first to study antibodies in milk by any means, but it's been more of like a general cross-sectional look at whatever antibodies of interest. And essentially nobody before we started doing our work, in 2020 was specifically looking at secretory antibody. Often it says secretory IgA, and then you read the paper and they're actually just looking at IgA and they claim it's secretory. The post-vaccine response, you will find IgA, but it's generally not in secretory form because it's coming in from the serum. And, vaccine response just don't last because they're more like the declining serum response. But there's something special about this mucosal response, this secretory antibody response that, I think we evolved to have these long-lived B cells in the mammary gland. We haven't dissected it. We don't really understand how this is such a great long-lasting response, but it definitely is. And we intend to look more at that. But that was definitely quite surprising, pleasantly surprising in our.
A good surprise to come out of a pandemic, maybe, but very interesting. And I think we do have time. I know we're at the half hour, but we have time for one more question. I think for you, Rebecca, could you comment on whether the antibody coating effect protection transferred through breast milk could be replicated in prepared formula?
Yeah, I just saw that question there. Possible. I think that, I don't know how, I'm not that kind of scientist, in terms of formulating things to work in that environment. I think that if enough research went into preparation of a formula, right chemical balance, that the antibodies could be maintained and not be degraded in those formulas. And I think there has been some attempt in certain formulas to include some kind of like immune boosting. I don't really know what's in them. I think it definitely could be researched. It's possible it could work, that formulas could have kind of like a total antibody content that's been extracted from like donor milk. You would want it to be in secretory form. Secretory antibodies are actually really, really difficult to make in the lab because they have so many components. So you can extract it from milk, and there are, we are trying to optimize with some collaborators how to best do that, but it's very hard to make large quantities of it. So you would want to extract it from a natural source like milk. And yeah, I think it's possible that you.
Well, that's very interesting. Thank you so much, Miranda, for that last question. And as we're wrapping up today, I do want to again thank all of you sincerely, all of our speakers, and to our audience members who joined us today. We really hope that we'll see you at our next event, which is coming up in April. So please keep an eye out for communications from our team. And again, thank you from all of us at the COVID Info Commons, and I hope that we'll see you in our next event.